Okay, right. So I got my connection dropped at some point yesterday towards the end. So if I cut off in the middle of things, I'm sorry about that. That was, some problem on my end. Let's let's get some of that glare out of the way. That might help a little bit. All right. And yeah, so let's go ahead and get started. And I'm going to recap what I was saying about the structure of proteins. So let's go ahead and kick this in. We were talking about proteins. Proteins are polymers of amino acids, and there are 20 genes. I mean, there's actually thousands of amino acids, all proteins, and proteins. We'll talk about one particular group of proteins, the enzymes, in just a bit. So the order in which you put amino acids together is a protein's primary structure. When you've put amino acids together in an order, some of those amino acids will start attracting or repelling other nearby amino acids. It's a little bit like static electrical cling or repulsion, not exactly the same, but that will cause the amino acid chain to fold up. And specifically, it will cause portions of the amino acid chain to fold up in various ways. Tertiary structure is caused by attraction and repulsion, again, kind of like static, between R groups, and that causes the entire chain, not just parts of it, to fold up. And then you can have several amino acid chains that'll fit together into a single molecule. So primary structure is just the order in which you string the amino acids together, one after another. This is kind of what it looks like. That's a simple amino acid polymer. They're representing nitrogen atoms in blue and oxygens in red, and that's just what this would look like. When nearby amino and carboxyl groups attract each other, regions of that chain of amino acids will start bending and folding. And there's two basic types of bending and folding. One's called an alpha helix, not the same as a double helix. Remember, a helix is any kind of spiral shape. So a portion of an amino acid chain will form a spiral. And then sometimes a chain of amino acids will kind of zigzag back and forth and form a shape called a pleated sheet, where the amino acid chain will run straight for a while and then kind of double back on itself and then double back again and double back again and do this kind of zigzag maneuver. So secondary structure refers to parts of that chain of amino acids folding up into alpha helices and beta sheets. Tertiary structure refers to the entire chain of amino acids, not just local parts of it folding up. This is a protein found in your muscles, and it happens to have a number of alpha helices. So the bits here that look like telephone cords are alpha helices, and then the parts that look like wires represent chains of amino acid that are not forming either an alpha helix or a beta sheet. And, they're just kind of zigzagging around. And the whole thing folds up into a tight, compact structure, one that, by the way, binds oxygen and transports it to your muscles to the spot where it's needed. So that's your tertiary structure. That second slide is the tertiary structure of another protein one around the other to give this kind of spiraling shape to it. Some are very rich in, okay, hang on, I heard a ding dong, so give me a second. So there's some that are made up of alpha helices and beta sheets as well. Okay, somebody has just joined us. Okay, good morning. We were recapping protein structure, and I got cut off somewhere on Friday. So I'm kind of giving you guys a recap. Probably doesn't hurt. Let me. Okay, I should be presenting, so I'm assuming everybody can see that. Most and some regions that make beta sheets, and then the entire bend around the sheets, or the sheets might wrap around the helices or something like that. This is one where you have alpha helices in red, beta sheets are in green, so there's kind of a tube made of beta sheets in the middle and a bunch of alpha helices wrapped around the outside. The parts of the protein that are neither helix nor sheet, they're in purple. And then finally, quaternary structure is what you get when you have several different chains of amino acids, several different polypeptides, each of which has its own tertiary structure, and they all join together. So in the case of hemoglobin, hemoglobin is the protein that transports oxygen in your red blood cells. It picks up oxygen at the lungs and transports oxygen to all parts of your body. And hemoglobin is actually made up of 4 different amino acid chains. 2 of them are a type called alpha globin, and 2 are a type called beta globin. And if you are making hemoglobin of the right shape, then 2 alphas and 2 betas pack together to make this very compact, very tight shape that's well adapted for carrying oxygens around. This is just another one where you have, you can see helices and sheets on both of these. One of these peptide chains is in blue and one is in green. There's another one. This is a protein that consists of 7 separate polypeptides, each of which is color-coded differently. So you have this kind of internal stalk of one polypeptide that's colored red, and then 6 more polypeptides that fit together around the stalk, almost like Trivial Pursuit pieces, giving a protein with a shape like this. The reason anybody would actually care about learning this stuff is, remember that protein shape is absolutely critical for protein function. Protein shape determines what the protein can and can't do. Your life depends on the fact that cells in your body can make thousands of different proteins, each of which is able to do something. If they did not do what they do, you would not do what you do, because you'd be dead. You wouldn't do, you'd die. That was alliterative. And you do what you do because your proteins do what they do, and they do it because they're shaped in a way that does something, much like tools are shaped for particular functions. I don't know if before we go on, I don't know if I got to this before we got cut off, but there's a couple of proteins that are found in wheat flour. And if you make bread dough and then you start kneading the dough, or your bread machine kneads the dough, what you're doing is taking those proteins and assembling them into a bigger and bigger and bigger and bigger quaternary structure, until at the end of your kneading process, you may have all of those proteins linked up in a giant molecular structure that fills the entirety of the dough. You're building a quaternary structure when you knead bread. By the way, those proteins are collective that you're building into this quaternary structure are collectively called gluten. If you have bread dough that you knead like that and you make that giant quaternary structure of millions of proteins sticking together, it tends to make the entire dough very elastic and very stretchy. So something like pizza dough, for example, holds together so that you can take the pizza dough and, spin it around in the air like they do. The reason you can do that is that that's a very high protein variety of wheat that's held together by that enormous quaternary structure of millions of interlinked proteins, making the whole dough very stretchy, very sticky, very, holds itself together very well. And that ends up with giving you a very, chewy, stretchy bread product. Varieties of wheat with less protein are used to make things like cake and pastry, which are more crumbly rather than stretchy, right? You don't eat stretchy cake. Your birthday cake is meant to crumble, which is why cake flour and bread flour are actually different from each other. And then, of course, if you can't eat gluten at all, you have to come up with varieties of flour that don't contain that protein but have other substances that can make the dough stretchy. And if you've ever eaten a lot of gluten-free products, you know that it's not quite the same. You can never quite get gluten-free bread that is as stretchy and chewy as, let's say, a proper pizza crust. And the reason for that is that depends on gluten. And the gluten in your bread dough is an enormous quaternary structure that you make by kneading the dough. Crumblier baked goods like cake and pastry and shortbread and things like that, you don't need those. They never build up that giant quaternary structure network, and they end up with a very different texture, a very crumbly texture. Right. So the point of all this stuff is that organisms make huge numbers of proteins. The proteins do huge numbers of things. They run your metabolism. They transport oxygen. They transport signals in your body. They hold you up. Your collagen and all that is made of protein. They glue your cells together. They enable your cells to receive signals from the outside. Everything is protein, protein, protein. Proteins can't work if they don't have the right shape, and the shape depends on the order of amino acids. If you put a protein together with the wrong order of amino acids, the protein will take the wrong shape, and then the protein won't be able to do what it's supposed to do. Incidentally, you can change the shape of a protein molecule without breaking it down. You can just make it take a different shape. Heat will do that. Acid will do that. Other chemical reactions can make a protein change its shape. That's called denaturing the protein. Simple example of denaturing protein would be frying an egg. The egg white is mostly protein, a protein called albumin. And when you fry an egg, you're not breaking the protein down unless you're burning it, but you're changing the egg from being liquid and translucent to being white and solid. That's protein denaturation. So is if you add certain bacteria to milk, the bacteria start breaking down the sugar, so the milk turns less sweet. And the bacteria secrete acid, which means the milk tastes sour. And the acid starts denaturing the milk proteins, which means the milk stops being liquid and starts getting chunky. And I've just described how you make yogurt. Oh, one more example of denaturing a protein. Your hair is made of proteins, a protein called keratin. And your hair has the texture that it does, straight, wavy, curly, what have you, because it's made of protein molecules of a particular shape. And if you change the shape of the protein molecules that make up your hair, you can change the shape of your hair shafts. And I've just described a perm, a permanent wave, is an example of denaturing the keratin proteins that make up your hair. You're not breaking the proteins down unless you set your hair on fire or something like that. I guess you might be if you really damage your hair. So always buy Max Factor products or whatever it is. Hope they pay me for saying that. But what you're doing is altering the shape of those protein molecules, and that lets you alter the shape of your hair shaft, which changes whether it hangs straight or forms curls and waves. So protein shape has a lot of things, of important things. Sorry, momentary distracted. I've got a cat trying to walk on me. Okay. Very, very, very important class of protein is a group called enzymes, and that's one you've probably heard of. What do they do? Starts like this. You store energy in the bonds that hold atoms together in a molecule, what we call chemical bonds. For our purposes here, we don't really need to go into what those bonds are. If you really want to know about ionic versus covalent versus hydrogen versus resonance and metallic bonds, we've got a perfectly good chemistry department that would just love to hear from you. For what we're doing in biology, we can just pretend that the bonds are little, popsicle sticks holding ping pong balls together. What you need to know is that atoms can stick to each other more or less permanently. It takes energy to make those chemical bonds, but energy is released when chemical bonds are broken. It's a little bit as if atoms were held together by little springs, and the springs were under tension. So if you cut the spring, the atoms will fly away going boing, boing, boing. It takes you energy to make something like that, but energy is given off when those chemical bonds are broken. So any kind of chemistry that you do involves breaking bonds between atoms and forming new chemical bonds. That means that a chemical reaction both needs energy and it gives off energy. If it gives off more energy than it consumes, the chemical reaction, once you start it, is going to keep going, and it's said to be exothermic. It's giving off more energy. Thermo means heat. If it gives off more heat than it requires, then it's exothermic and it will keep on going. And the simplest example I can think of is fire. When you set something on fire, whether it's a piece of paper or a log of wood in the fireplace or the body of someone who cheated on one of your exams, not that I ever did that. Once you get that fire started, you don't have to keep adding energy to it. It will just keep on burning. What you're doing is breaking the bonds that held together the carbon, hydrogen, oxygen, and nitrogen atoms that made up your wood or your paper, let's say. You're breaking the bonds that held those together, and you're combining those atoms with atoms of oxygen from the air. The whole thing gives off more energy than it requires. So once you have struck the match and lit the fire will keep on going until there's nothing more to burn. If that reaction consumes more energy than it produces, then you have to have a source of energy to proceed. A lot of cooking is like that. You do chemistry when you cook anything, and to cook something, right, you can't just, put your roast in the oven and just stare at it until it spontaneously browns and gets tender. You have to keep adding energy until the reaction has gone as far as you want it to. And a reaction like that would be endothermic. An endothermic reaction consumes more energy than it gives off. An exothermic energy gives off more energy than it consumes. So you can graph the amount of potential energy in a chemical system over time as something like this. When you have, let's say, a bunch of oxygen and a bunch of wood ready to start the fire, there's energy stored in all those chemical bonds. Once you start it burning, that energy will be given off until, if the burning is complete, you end up with nothing but carbon dioxide gas and water vapor. I mean, usually there's unburned things like, ashes and smoke. But once that reaction goes all the way down, you have less energy in the products than you did when you started. You had stored energy in the wood and the air when you began the burning. And at the end, when all you have is steam and CO2, you have less energy in those than you had when you started. So you can think of the reaction as running downhill, right? Once you start a ball rolling downhill, it will keep on rolling until it gets to the bottom. But there's something called activation energy that we have to account for. You can put a bunch of wood and paper in your fireplace and then just sit there and stare at it, and it is never going to catch on fire itself because you have to add a little bit of energy to get that ball rolling, right? You have to strike a match, right? You have to apply a little bit of energy to get the fire started. As we used to say in my church back in the 70s, it only takes a spark to get a fire going. And then we'd sing Kumbaya. Anyway, and in this kind of drawing, we can draw this as a little hump. Imagine you've got a ball at the top of a hill, and right in front of that ball, there's a little speed bump. Once you get that ball over the speed bump, it will roll downhill until it gets to the bottom. But you have to give it a little kick to get it over the speed bump before the ball will start spontaneously rolling. That little kick that you give it corresponds to what we call activation energy. It's the energy you have to add to get an exothermic reaction started. There's a class of proteins called enzymes, and what they do is decrease the activation energy. It's the equivalent of coming in and lowering that speed bump until the ball can spontaneously roll downhill. In fact, any substance that lowers the activation energy of a chemical reaction is called a catalyst, and enzymes are proteins that act as catalysts. A catalyst isn't burned up by the chemical reaction. A catalyst will keep on working for as long as you care to. In your cars, at least if you drive something built since the mid-70s, which I assume most of you do, there is a gizmo on your exhaust manifold called the catalytic converter. The catalytic converter takes some of the hot gases that are coming out of your car, like carbon monoxide, and does some chemistry on them and converts them into things like carbon dioxide that are a bit less dangerous. It does it by using platinum as a catalyst. Your car's catalytic converter has tiny amounts of platinum in it. That platinum is not burned up by the reaction, right? You don't have to go in for periodic platinum refills, which is a good thing, or maintenance would be a lot more expensive than it is. That's because catalysts are not consumed by the chemical reaction. Enzymes are proteins that act as catalysts. Incidentally, I won't say much more about them, but there are some small molecules that aren't proteins, but that play helping roles in making chemical reactions happen. We call these coenzymes. And some, although not all, of your vitamins turn out to be coenzymes. They aren't proteins themselves, but they do various things to help certain chemical reactions along. They work with enzymes, if you want to think of them that way. So if an enzyme is present, it drops the activation energy. That makes it much more likely that the reaction will proceed all the way to completion. I'm trying to think of, oh, here's an example you could try at home. Okay. Jello is flavored protein. It's actually flavored collagen protein that's powdered, and then when you mix it with hot water and then let it set, it chills into this wobbly protein network. That's basically what you're eating when you're eating Jello. You're eating collagen protein with fruit flavor. Some fruits cannot be put in a Jello salad. You know what a Jello salad is, right? That is pieces of maybe canned fruit mixed in with your liquid Jello and then allowed to solidify. So you have, pieces of fruit embedded in the Jello. I'm assuming you've seen things like this before. One of the fruits that you cannot put in a Jello salad, in a congealed salad, is fresh pineapple. And the reason you can't put fresh pineapple in a Jello salad is that pineapple and a couple of other fruits, kiwis are another one that come to mind, and I think mangoes do this too, they contain an enzyme, and it's an enzyme that breaks down collagen and other proteins. It's called papain. And so that papain enzyme, if you add fresh pineapple to Jello, your Jello will never solidify because enzymes in the fresh pineapple will break down the collagen. And you'll end up with gooey Jello instead of Jello that sets up and forms a nice network of solid of a nice network of protein molecules. Canned pineapple should work because canned pineapple's been heated, meaning that the papain enzymes should have been denatured, which means that they're no longer able to break down the collagen proteins in Jello. The enzymes don't work because they've got the wrong shape. I ought to try that this afternoon. I might go out and get me some fresh and canned pineapple and show you how this works. This would actually be an interesting demo. I'm glad I thought of that. Yeah, you can actually do some household demonstrations of how this works. Right. How does all this work? Well, remember that protein shape is critical here. The molecule that an enzyme acts on is called the enzyme's substrate. And an enzyme molecule has a single region on it called the active site, and the substrate has to fit into that substrate like a key fitting into a lock. Or if you'd rather, you could think of, if you want to think of enzymes as being like tools, let's say the active site would be like the head of your screwdriver or the socket on your socket wrench, in that whatever that your tool is supposed to work on, the screws or the bolts or whatever it is, the screws and the bolts have to fit the head of your screwdriver or the socket of your socket wrench. They have to fit it very precisely. The rest of the tool may not make that much difference. It may not matter very much how long the handle of your screwdriver is, or what color it is or anything like that. But what matters is that tip of the screwdriver, because that has to fit the screws that you're working with. So you could think of the screws as being like the substrate, and the screwdriver as being like the enzyme. And the enzyme has a single region, the active site, that's like the point of the screwdriver. And the substrate has to fit into that like a key fits into a lock. Once the substrate molecule is fit into the active site, the enzyme molecule might put stress on it. The enzyme molecule might bring it next to another molecule that fits, and the substrate will either break up or the substrate might come together with something else. So if you want a diagram of it, up at the top left, you have that green thing. That's a molecule that acts as a substrate. The green thing fits into the active site. The active site changes shape a little bit to fit the substrate. That's called induced fit, and we don't need to worry about that. But the enzyme may put just enough stress on that substrate that substrate will break apart. In other cases, you might have the enzyme inducing the substrate to form a bond with another atom. So enzymes can build up and they can break down. And as I've said, you might compare an enzyme to a tool like a socket wrench. The length of the handle or something like that may not make a huge amount of difference, but what's absolutely critical is that socket. The socket is the active site, and that active site can fit only one size and shape of substrate, which in this case would be the nuts or the bolts, whatever you're trying to loosen or tighten with your socket wrench. Here's a real world example. This god-awful thing is an enzyme called beta-lactamase. The green areas here are alpha helices. The red areas are beta sheets. And then the blue wiry looking stuff are regions of that chain of amino acids that are neither helix nor a sheet. And then the thing to the left that looks like a little yellow bundle of sticks, that's a model of a molecule of the antibiotic penicillin. And when a penicillin molecule fits right there into the active site on beta-lactamase, beta-lactamase puts strain on it and causes it to break apart. The reason this matters is that bacteria that can make beta-lactamase enzymes are resistant to penicillin because they can break it. If you zap those bacteria with penicillin, penicillin would normally block those bacteria from producing cell walls, but these bacteria can break it down. Some of them can even break it down and eat it. There are bacteria that can use penicillin for food. And this is one of the many ways in which bacteria can become resistant to antibiotics, is that they have evolved by our old friend natural selection enzymes that are very good at breaking down penicillin. And humanity has to keep developing new antibiotics as the old ones become less and less useful because bacteria evolve enzymes that can break down the older antibiotics. So there, that's a callback to natural selection there. We talked about antibiotic resistance earlier. And you could think of metabolism as being like an enormous set of assembly lines building up and breaking down molecules, and every enzyme would be like one of these machines carrying out exactly one step, right? The car chassis right here goes to one machine that goes zip, 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 maybe, tightens 4 bolts or something. And then that chassis moves to the next machine, which goes zup, 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 and tightens 4 more bolts. And then it moves to the next machine, which, brings the doors in and goes zip, zip, zip. I don't know how this works. I have never built cars, but every enzyme would be one of these machines carrying out one and only one step indefinitely, 24/7. It's a little more complicated than that because the assembly line here is actually forked. Maybe some of these cars are destined to be, have V6s, and some are destined to have V8s. So some of these car bodies will go off in one direction and some will go off in another direction, right? You can fork an assembly line. Well, the assembly line of metabolism looks like this, and I hope you copy all of this down. Actually, I'm kidding. Please do not copy all of that down. But all of those thin black lines that you can see is one chemical reaction. Every one of them requires a specific enzyme to make happen. So every cell in your body, well, there's a few here that are photosynthesis reactions, which your body doesn't do. But aside from that, every cell in your body has to create an enzyme for every one of those little black lines. Those are steps in the metabolic assembly and disassembly line. Every one of those black lines corresponds to an enzyme that has to be made with exactly the right shape. I'm imagining you all just going, ooh, ah, your mind should be completely blown by now. Here's a tiny piece of these reactions. By the gods of Valhalla, don't memorize this stuff. What you need to know is they're a tiny little part of the reactions. If we go back to the whole thing, those reactions are... I can't even read this really well. Those reactions are kind of somewhere. Okay, they're kind of up in the center, shaded in pink. If you see a little vertical area that's got some pink shading kind of in the middle of the diagram, this is where we're talking about. They look like this, and they start with a simple sugar called glucose. And glucose is broken down into glucose 6-phosphate, which is turned into fructose 6-phosphate, which is turned into fructose 1,6-bisphosphate, which is broken down into GLAP and DHAP. DHAP's turned into GLAP, and then GLAP is turned into 1,3-bisphosphoglycerate, which is turned into 3-phosphoglycerate, 2-phosphoglycerate. Okay, don't worry about that. Okay, that's what? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different steps that break down glucose sugar into a simpler compound called pyruvate. Every one of those steps is catalyzed by an enzyme, and the enzyme names are shown in red, except for one that's shown in green. So to do all of that, you have to make 10 different enzymes, each of which has a particular shape to it. There's a species of yeast called Saccharomyces cerevisiae. You might remember that yeasts are fungi that don't form mycelia, or at least they don't all the time. And yeast is cool because if you give it sugar, it will turn glucose sugars into pyruvate, and then it will take the pyruvate and feed it into another set of chemical reactions, each of which is driven by one enzyme, and that's called fermentation. And so yeast will turn glucose into pyruvate and then ferment pyruvate into ethyl alcohol, which is the type that some people drink, although this being Faulkner County, I have to say that not everyone does. And a byproduct is carbon dioxide. So there's several steps by which pyruvate is broken down into ethyl alcohol and carbon dioxide, and every enzyme controls one step in the process. So if you take some Saccharomyces cerevisiae, seen here under the microscope, the actual size is maybe 10 microns or so. Oh, and incidentally, when they divide, they don't divide evenly into two equally sized cells. They divide into a main cell and a bud, which is what you're seeing there. You're seeing a couple that have just divided into a main cell and a bud. And when you turn these yeast loose on a water solution that's got lots and lots of sugar in it, the yeast will break down the sugar into pyruvate and then convert the pyruvate into ethyl alcohol and carbon dioxide. If your sugar solution is grape juice, the yeast will convert your grape juice into wine. Usually the carbon dioxide is allowed to not evaporate, but is allowed to bubble off. So most of the wines that you drink are still wines. But if you trap some of that carbon dioxide, there's a special method for doing this. If you trap some of that carbon dioxide in the bottle, you get sparkling wine. You get champagne or spumante or sparkling wine. That uses both the alcohol and the carbon dioxide to make the finished product. So Saccharomyces cerevisiae turned loose on grape juice makes wine. Turn it loose on another fruit juice, and you might have a fruit wine, strawberry wine, like that old country song, or, 100 gallons of sweet red wine made from the biggest watermelons on the vine. That's another old country song. If you turn it loose on a solution of honey and water, you'll make mead. If you turn it loose, there's my wife leaving the house. Have a good time, sweetie. I love you. I love you too. All right. If you turn it loose on rice that's got another fungus growing on it, you make sake. And then you can also turn it loose on a grain called barley, which looks like this. Barley does not have much sugar, but it has got lots of starch. And you might remember that starch is a polymer of sugars. So you can soak barley in water and then spread it out, keep it nice and warm, and let it dry, which is what this guy is doing right there, spreading out barley grain after soaking it. And each seed will just begin to sprout. And when it does this, it's called malt. So barley that has just begun to sprout, not enough that you can see little plants coming out of the seed, but just beginning the process, that's called malt. The process that he's doing is called malting. And what malting does is make those seeds start producing enzymes that start breaking down the starch. Starch is a polymer of sugars, and the barley will begin to produce, the seeds will begin to produce enzymes called amylase, and the amylase will start chopping up these starches into a simple sugar called maltose, which the seed would normally use as an energy supply. The starch is there to give the seed enough energy so that it can grow into a plant that's big enough to start doing photosynthesis and generate its own. The seed is that energy, the starch in the seed is the energy source needed for those early stages of growth. So if you get the seed just beginning to grow, the amylase will turn the starch inside the seed into sugar. And once you've done that, you can take the barley, grind it up, soak it in hot water to create a sweet extract. If you must know, it's called the wort. And you cool that hot extract down and you feed it to Saccharomyces cerevisiae, the yeast, which will use its own enzymes to break the maltose into glucose and then break the glucose through many steps into ethyl alcohol and carbon dioxide, and you've got beer. Traditional beer retains the alcohol, and it also retains some of the carbon dioxide from fermentation. Mass-produced beer actually has CO2 pumped into it to make it more fizzy. But if you've ever had what they call cask-conditioned ale or bottle-conditioned ale, home brewers do this a lot. If you've ever had somebody's really good home brew, the bubbles in your homebrewed beer are carbon dioxide left over from that fermentation process. So enzymes are extremely important in creating beer, wine, any of those drinks. Not that I'm allowed to encourage you to sample any of these because we are in a dry county, of course. I've actually had people ding me on evaluations before because they thought I was teaching people how to make alcohol and, leading them down the path of sin. So I certainly do not want to interfere with anybody's choices on that matter. But ultimately, this is how you make this stuff. And ultimately, this is one of the things that you need to know about enzymes to do. Home brewers can get pretty technical describing the exact process that they use in order to get the flavors that they're trying to get in the beer that they make. And they'll talk a lot about, maltase to starch conversions and things like that, because they're using knowledge of, enzyme activity in order to design the beers that they make. Incidentally, there's a really good homebrew store called Fermentables in North Little Rock if you ever get interested in this sort of thing. And not that you asked, but yeast get poisoned by alcohol once you get that alcohol concentration up to like 18% or so. So if all you use is yeast to make your alcohol, you'll never get anything stronger than about 18%. That's 36 proof. This stuff, which is 92 proof, or 46% alcohol, is made by brewing a beer or a wine and then distilling it to concentrate the alcohol, heating it, collecting the alcohol vapors, and getting rid of the rest. So by doing that, you're concentrating the alcohol, making a product that is more alcoholic than anything that your yeast can make for you. And often you'll store this in a wood barrel for some time because that mellows out the flavor. So this bourbon whiskey will have been aged for 3 years in a proper oak wood barrel. So that's how you make brandy, whiskey, vodka, anything like that, anything that's significantly stronger than beer or wine. Or cider. If you zap apple juice with yeast, you get cider. I forgot about cider. Okay, that's actually a pretty good place to take a break. It is 10. So I'm going to go black screen to go away. And okay, I think we've stopped presenting there. And I'll go ahead and stop recording.